Good evening. I'm Sandy Landman from the Friends of the East Brunswick Environmental Commission, and I'd like to welcome you to the first Option Green program of the new season. We're virtual tonight. So I'll tell you first a little bit about our group, and then we'll get started with our great program. If you're not familiar with the name of our group, the Friends of the East Brunswick Environmental Commission, you're probably familiar with our programs. Because we sponsor many educational, environmental, and fun community programs here in East Brunswick throughout the year. And they include the East Brunswick Community Garden, which is right near the municipal complex, the Salamander Protection Program, which is really popular, and that gets underway in the kind of middle of the winter. National Moth Week, which is an international citizen science project that started right here in East Brunswick 10 years ago in 2012. And now it's an annual observance all over the world. We also sponsor local moth nights, which we had back in July. We work with the library on the seed library, which is right now dormant for the season and will reopen again next spring. So that's a great place to find and donate seeds. And now something new, we're partnering with the new Sustainability Task Force, which is working on reducing the use of single-use plastics in our schools. And I think you'll be hearing more about that in coming weeks and months. Some good news, Free Cycling Day, which everyone in town looks forward to, is going to return to the Community Arts Center this year on Saturday, October 8th. Please watch for more details on our website, which is friendsebec.com, and Lidi Haramati of the Friends is going to put that into the chat, so you'll have that link. We need volunteers, so we'll be posting a link to a form. So if you'd like to volunteer for a shift at Free Cycling Day, it's a fun day to work and see all the things that people are bringing to recycle or free cycle. And lastly, before I introduce our speaker and our program, I just want to let you know that our next Option Green program is one that you won't want to miss if you live in East Brunswick. Dan Lozick, who's the Director of Water and Sewer Utilities in the Department of Public Works, is going to talk about water quality in East Brunswick, and how they protect it, measure it, etc. And that talk is coming up on Wednesday, October 19th. It will be hybrid. It will be at the library and online. So we hope you'll be able to sign up or just show up for that one. And we'll be posting those details very soon. So now on with tonight's program. We're very pleased to have marine biologist Austin Grubb of the Rutgers Department of Marine and Coastal Sciences with us. Austin is going to tell us all about microscopic phytoplankton. I hope I'm saying that right, Austin, and why we should all know about and care about them, because despite their tiny size, they play a very big role in the ecosystem of our whole planet. And he'll explain what that means in his program, in his talk. Austin will tell us how oceanographers study phytoplankton in the environment, and he's going to talk about his recent research cruise, and I think show us a video, right, Austin? You'll be able to put questions into the chat, and when he's finished with the talk, we'll take your questions, and I'm sure he'll be happy to answer them. So Austin, you're on, and enjoy Austin's talk.
Thanks for the introduction. So as Sandra said, I'm Austin. I'm a PhD candidate in oceanography at Rutgers University, and I'll be telling you about microscopic organisms with global impacts, how phytoplankton impact the Earth's carbon cycle. And so don't worry, I'll start with some background info about why everyone should care about carbon to begin with, and then introduce the carbon cycle, lead in and say a little bit about phytoplankton before diving into more specifics about my work and why that's important and what I have found. And then I'll end with with some videos and pictures and other details about the recent research cruise I was a part of. But first, I thought I would take a little bit more time to say a little bit more about myself and my scientific journey, since I'm sure not everyone comes into contact with scientists very often. And so I am originally from South Central Pennsylvania. And since about middle school, I knew that I wanted to study to become a marine biologist because, like I'm sure many kids, I really like dolphins and whales and sea turtles and other animals in the ocean and cared about protecting them and everything. And so by high school, the time to start thinking about colleges and applying to colleges, I had the end goal in mind, but I did not know what the path to get there was like and what I should plan on. I'm the first person in my family to go to college, so it's not like I really had many people around me in my family or scientists I could go talk to in my community to get advice from. So, like all young people today, I turned to the internet and was able to gather from the internet and also from guidance counselors and such that I could major in biology in college and then specialize in marine biology later on in graduate school, which is what I ended up doing. And so that led me to Susquehanna University, where I completed my undergraduate degree in biology. And I chose Susquehanna University in part because they had a strong freshwater ecology research program. They are named after the river in central Pennsylvania, after all. And I thought that I would be able to get some good research experience in a freshwater ecosystem that then I could potentially apply to the marine system later on in my career. And so I quickly learned, though, at Susquehanna, that I was not interested in fish research. So the most of the research around fish at Susquehanna was about managing them as a resource, and that just wasn't something that sparked my interest that much. And I also learned that I was not very interested in benthic macroinvertebrates or aquatic insects, one of the other main areas of freshwater research that was available. I think a person can only pick so many bugs out of a tray of preserved bugs and alcohol for 8 hours a day, every weekday for so many weeks until you decide that that's not for you. And that's what I decided. And so the other big area at SU of aquatic research in terms of on the biology side of things was microalgae. And so I had some knowledge of microalgae from some classes. And then I took a class called aquatic ecology, and they intrigued me. They're small microscopic organisms, so you can't see them with your naked eye, but they have large impacts on the ecosystem. They form the start of food webs and such. And so that ultimately led me to a summer research opportunity, an internship at the University of Rhode Island's Graduate School of Oceanography. So these are, and specifically a research experience for undergraduates that are funded by the National Science Foundation, or the NSF. And so during this 10-week internship, it was my first real exposure to oceanography, and I got to work with phytoplankton. And so I worked with this phytoplankton, which is a diatom, and I'll talk more about those later. But part of the project ended up being growing them in these big tanks with these bars that would move up and down to try to mix the tanks and to prevent them from settling on the bottom here. And I quickly learned that 10 weeks is not long enough to finish a research project. So even though I unfortunately wasn't able to complete this project, it's the whole experience, the lectures that I attended, and just the research in general hooked me on oceanography and phytoplankton specifically, and I knew that I wanted to study them. So then back at SU for my senior year, I ended up working with on benthic algae, which are just algae that grow on surfaces such as rocks on the bottom of stream beds and riverbeds. And so here in this photo, I was preparing clean rocks that are in this like metal gasket thing, along with some glass microscope slides that then we placed in the Susquehanna River for a few months. And this just gave time for the rocks that were clean again to be colonized by benthic algae that were in the river. And then we would bring those back into the lab, clean the rocks, basically kill anything that's on the rocks, and we'd be left over with the shells of these algae. And these are also diatoms, but they're just diatoms that grow on surfaces rather than in the water column like phytoplankton do. And I spent the rest of my time during my senior year identifying and counting diatoms from these samples. And so then that data was used as bioindicators of water quality. And so these diatoms can be used as that because aquatic ecologists have determined that certain species prefer different conditions. So when you have a lot of species that tend to exist under certain conditions line up, you're able to match, okay, the water quality here, according to the diatoms, is pretty good, or, oh, it's not so great because these diatoms tend to do well in dirty areas, but the ones that do better in cleaner waters aren't prevalent. And so that was my senior research at SU. And so then I applied to graduate school programs, oceanography programs, and I ended up at Rutgers, which is where I'm at now in the Department of Marine and Coastal Sciences. And I've been there ever since. I'm just at the start of my 6th year as a PhD candidate. And now I study this group of phytoplankton, which are called coccolithophores. I'll get more into these guys later, but first, I'm just going to provide everyone with more background information. That way, hopefully, you'll be able to follow along with my research and why they're important. So first, why do we care about carbon? So I'm sure many of you may already have an idea of why we should care about carbon, and specifically carbon dioxide. But we care about carbon because carbon dioxide is a greenhouse gas. And we know that greenhouse gases trap solar radiation or sunlight that enters the Earth's atmosphere and traps that as heat and prevents that sunlight from being reflected back out into space. And this is the greenhouse effect, and this warms the atmosphere, and so it warms the surface of the planet. The greenhouse effect alone is not bad. The surface of the Earth would not be able to support life as we know it without the greenhouse effect. It's just that human industrial emissions have increased greenhouse gases like carbon dioxide high enough that now we are increasing the greenhouse effect and thus warming the planet. And that's global warming in a nutshell. And so here is a video that shows the increase in carbon dioxide levels in the atmosphere. So there's the year on the bottom here, and the CO2 levels in the atmosphere is on the left here. So you can see that it's increasing over time. And these are at 2 different stations. The red line is in Hawaii, so that's the northern hemisphere. The blue line is at the South Pole, so the southern hemisphere. And you can see it oscillates up and down. The general trend is up though, and that oscillation is due to the seasons, which is all driven by photosynthetic organisms like plants. And now the video will just back up a bit to backfill in data. And then now we're going to start to fill in data from ice cores. That way we get a sense of what pre-industrial CO2 emissions were like. So right around 280 or so, whereas now we're over 400. This video stopped at 2014. We're higher than that now. And now if we just go back tens of thousands of years, eventually hundreds of thousands of years, we can start to see the cycle of CO2 levels in the atmosphere over recent geologic history. And let me pause it, that way it doesn't continue. And we can see there's a very natural cycle of carbon dioxide levels in the atmosphere. And then a not so natural spike here at the end that is way out of bounds compared to what came before. And that spike is the emissions from human industrial activity. And I'll just play the end of the video. That way all of the groups that contributed to this can get their credit. Right, and so this image goes up to 2021. So we're now well over 400 parts per million of carbon dioxide in the atmosphere. I think we're somewhere between 415 and 420. And this has led to a warming of the Earth's atmosphere. So we're now at over 1 degrees Celsius of warming. You'll hear different numbers depending on what the initial baseline is. So this is an early industrial baseline. Some baselines go back a bit before that. But if you ever follow the International Climate Accords, the goal is to limit warming to 1.5 degrees Celsius, because warming past that would be pretty catastrophic. And we're already dealing with warming that's above 1 degree Celsius, maybe at somewhere between 1.1 and 1.2, depending on when you start to count. So that's why we should care about carbon in the atmosphere, because it can lead to bad things with climate change, of course. And as I mentioned in the video before, plants through photosynthesis have a big impact on that by taking up carbon. So all plants perform photosynthesis, and that's where they use sunlight, carbon dioxide, and water to produce sugar that they use for food, and that they use to make up their structural components that ultimately make up their organic matter or their biomass. And then they also release oxygen as a result of photosynthesis. And we're really lucky that they release oxygen, because as animals, we need oxygen in order to breathe. So without photosynthesis, we would not be in great shape. So that is photosynthesis. And so now if we plug photosynthesis into the carbon cycle, we'll start again with photosynthesis and see that plants take up carbon dioxide and they lock it into their bodies. And but they also respire just like we do. So they breathe, which does consume some of the organic carbon that they fix through photosynthesis, but they still take up more carbon than they release. And they are also consumed by animals, and animals also breathe and release carbon into the atmosphere. And of course, all living things eventually die, and that organic matter, whether it's from plants or from animals, will decompose. And through decomposition, the organic matter that locks up what was carbon dioxide can then be released back as carbon dioxide into the atmosphere. And some small portion of it, though, will be incorporated into the rock record as fossils and fossil fuels. And of course, the extraction and burning of fossil fuels is what is contributing to the increase in CO2 in the atmosphere. So now I have another video where you'll just be able to see the seasonality of CO2 levels in the atmosphere, as well as plant growth across the globe. So we're here in the northern hemisphere. There's more land here, so you're going to see more plant growth and higher CO2 levels compared to the southern hemisphere. But the pattern is the same in either, just the reverse, since the seasons happen at opposite times. And so the cutoff here for the CO2 level is about 374. So that's where you'll see the lightest color that's already here. Wherever there's a no color, it just means it's below that, not that there's no CO2. And as the colors get darker towards this orange and almost red-orange color, that's where the higher CO2 levels are. And then for the plants, where it's greener, as in darker green, there's more plants there. And so you'll be able to see, once I hit play, that as spring and summer comes, so that here's the time here, plants will take up CO2, reducing CO2 levels in the hemisphere. And then winter, more emissions are released, increase in CO2 levels, but then plants will draw it down. And again, the same things happening in the southern hemisphere as the northern hemisphere, just at opposite times. Right, and so this is in part what's responsible for that oscillation in the first video I showed you, where even though CO2 levels are still going up over the course of a year, they will fluctuate because plants will help draw them down. So here's another representation of the carbon cycle. So plants perform photosynthesis, lock carbon into their bodies as biomass. They also will breathe out some carbon. Some carbon is going to be stored in the ground and in soil. Microbes will help release carbon that's in the soil and such. This one leaves out animals, but they have their same role. And then, of course, human emissions. But this leaves out half of the carbon cycle. I'm sure by now you can guess that the other half happens in the ocean, based on the title of my talk. And basically similar processes happen in the ocean. There are phytoplankton that perform photosynthesis and take CO2 and put it into their cell matter or their bodies, and that is organic matter or organic carbon then. And then there are microbes that will respire them, that carbon, and decompose it. And of course, other animals that will eat the phytoplankton, and then animals that eat them. And some carbon can also be locked away if it sinks out of the surface ocean into the deep, and maybe even make its way towards the sediment. So now here's another video. And so in this one, it's similar to the last one, except now it's only focusing on the photosynthetic organisms. So if it's green, it's something that does photosynthesis. So on land, it's land plants. In the ocean, it's phytoplankton. And so there's no indicator of CO2 levels anymore. And here you'll see how basically the other half of the carbon cycle filled in from phytoplankton. And you'll see there's seasonality in the oceans as well. So see now the northern hemisphere is very green all over, and then it shifts to the southern hemisphere and the Southern Ocean. It goes back north, and then fades and goes south, and just repeats that. There's no time here, but you're just seeing months of data compiled. And it's just one way that you can watch the Earth breathe, basically, right? Because everything that's green is taking up carbon dioxide and is releasing oxygen. All right, so I've mentioned them enough already, so I should get around to actually telling you what phytoplankton are, I guess. So plankton are any living thing that cannot swim against a current. So they are free drifters. And phytoplankton are simply plant plankton. There are also animal plankton or zooplankton, some people say zooplankton, that will then feed on phytoplankton. And so phytoplankton are found in marine and freshwater ecosystems. And as I mentioned before, they're responsible for about half of the photosynthesis that happens. So land plants are responsible for about half, phytoplankton are responsible for the other half. And what this means is that every time you take a breath, half of the oxygen that you're breathing in is from phytoplankton, the other half is from land plants. So without phytoplankton, animals wouldn't exist. And despite, though, there being an almost 50-50 share of photosynthesis happening, phytoplankton have a lot less biomass. So if you were to take all the plants in the world and all the phytoplankton in the world and somehow be able to measure them against each other, and there have all of the photosynthesizers in the world, phytoplankton would represent only 1% of that biomass. Yet despite this biomass, since they are so small, they still do half of the photosynthesis. And as with plants on land, phytoplankton are the base of marine food webs. So the food web will start with phytoplankton. They will be consumed by zooplankton, which are then consumed by other animals, and everything dies and will be decomposed, in part by bacteria, which will release carbon in the end as well. And phytoplankton come in many different shapes and sizes. They're very diverse. They're actually way more diverse than land plants. Land plants are actually all descended from one group of phytoplankton. And so since phytoplankton are microscopic and you can't see them with your naked eye, there is this image here that scales up the size of phytoplankton cells to the size of things that we can see with our naked eyes. So on the smallest end, we have a fish representing the smallest phytoplankton. And over on the right, we have a representation of the largest phytoplankton, which is Manhattan. So that's quite the size range. The species I study is, let me get the laser pointer, is Emiliania huxleyi. So that's right here. And so I guess according to this graphic, I study the orca of phytoplankton, which I will take. And so now to show you some real cool images of phytoplankton, we'll start with this group. So these are all light microscope images. And these are diatoms. And so diatoms are phytoplankton that form shells made of silica. And so it's often said that they are algae that live in glass houses. And you'll see even among diatoms, there's plenty of different shapes and sizes. Some diatoms form chains. So the ones I studied in at Rhode Island for a summer are chain formers like this. There are other types of chain formers, and they're just really cool to look at. And they were used in some like 1800 in art from the 1800s too. Moving on to some other groups. Over on the left here, both of these images show the same group. So the top is another example of images from a light microscope. These are all cells of the same species of coccolithophore. And on the bottom here are just scan and electron microscope images of different coccolithophore species. So for the scan and electron microscope, rather than using light to observe the subject, we use a beam of electrons that's then converted into an image that we can see and this allows us to see things that are smaller at higher resolution. And so coccolithophores also form shells. However, unlike diatoms, their shells are made out of calcium carbonate or chalk. So you could say that they are algae that live in chalk houses rather than glass houses. The species that I study is this small one here in the middle, E, Mainania huxleyi, as I said before. And despite being one of the smaller species of coccolithophores, they actually are the most abundant in the ocean, and they're found almost everywhere in the ocean. Pretty much everywhere aside from the most polar regions. However, they seem to be going towards the poles as the oceans warm. And then there's some other groups represented on this slide. So we have some dinoflagellates that are just really freaky, a really freaky group that come in different shapes and sizes and have really complex life histories. Here's an example of a silica flagellate. So another example of one that incorporates silica into their body. And then the ones on the bottom here are the green algae. And they're just different examples here of them. So all land plants are actually descended from green algae. So all land plants come from this group. Lastly, I'll just mention the cyanobacteria. So these are bacteria that are phytoplankton that perform photosynthesis. They tend to be on the smaller end of things, and their direct ancestors were the original phytoplankton. So all organisms continue to evolve, none stop changing, but these would be the direct descendants of the first phytoplankton. And you can see that they're pretty small compared to some of the other images, and lots of them form chains as well. And so we actually have an example here of one of the smallest ones. So this was the fish in the graphic from a few slides back. So on the smallest scale, it's an individual cell of the cyanobacteria. And then over here on the right is the largest one that you can actually see with your naked eye a little bit. And that's simply because they form these really long chains that, that clump together and form these mats. And that's why they can be so large and can be the Manhattan of phytoplankton. There we go. All right, so I mentioned that phytoplankton are important to the carbon cycle because they do photosynthesis. But another way that they impact the carbon cycle is that they facilitate the biological pump and the export of carbon from to the deep ocean. So what does that mean? So if we look at this image, we see there are phytoplankton near the surface of the ocean, which they need to be since they need light to perform photosynthesis. So they will be taking up carbon and also respiring some carbon, but tend to take up more carbon than not, after all. And then they will be consumed by the animal plankton, so zooplankton, which are then eventually consumed by other animals. And phytoplankton can also be infected with bacteria and viruses. And so between the viruses and bacteria popping phytoplankton open, and between the zooplankton being messy eaters and leaving crumbs of phytoplankton floating around in the surface layer of the ocean, there will be many bits of dead phytoplankton cells, as well as other pieces of other dead organisms floating around. And this would be called detritus. And these particles can clump together to form larger and larger particles and form aggregates. And these particles can then sink better clumped together since they're larger and probably have greater mass than if they were not clumped together. And so these can sink out of the surface ocean to the intermediate zone and eventually to the deep ocean. Along the way, bacteria and other microbes like archaea are going to be consuming the particles on these aggregates and will be decomposing, helping to decompose the organic matter. So they will be releasing carbon back into the seawater. But fair amount, but some of the particles, some of that organic carbon will reach the very deep ocean, and a small fraction of it will eventually reach the seafloor to be buried and be incorporated into the sediments. So because of phytoplankton, there's a direct connection between of organic carbon from the surface of the ocean to the deepest parts of the ocean. All right, and so phytoplankton, when found in high enough numbers in a region, can actually be seen from space. And so these are blooms, as we call them, that can be seen from space. And so this is off the coast of the mid-Atlantic. So there's Virginia, Maryland, Delaware, and New Jersey here. And this color here is the phytoplankton bloom that was observed by this satellite. And so we know it's a phytoplankton bloom because when light hits the cells, the light will be affected in such a way that it's reflected back out of the ocean and changes the color of the water. And so that's when we know that it's a phytoplankton bloom. And again, this only happens really when there's a lot of phytoplankton there. And so I have a few more examples of blooms here. So here is one in the Gulf of Finland in Europe. And so you can see there are different colors here that probably indicate they're just different species represented than in the previous picture. And this is almost like putting food coloring in a glass or in a pot before you stir it. You can see how there's the swirl of the waters. That's probably from eddies and other physical properties of the ocean, currents and such. Here's another image of a bloom that's off the coast of Argentina. So off the coast of South America. And again, there's some different colors. And then here's another one that is in the Gulf of Mexico. So this is Texas, and then Louisiana, the Mississippi Delta, where the Mississippi River dumps into the ocean, is right here. So that's what all this tan coloration is from the sediments. And there's also the phytoplankton bloom. And these are all pretty much from NASA. And if you really wanted to, you could spend a lot of time going through all of the really cool satellite images that NASA has of different phytoplankton blooms from across the world. And I spent a fair amount of time selecting pictures for this talk. And if we focus back in on the coccolithophores, so the phytoplankton that I study, they can be seen from space even better than other phytoplankton because of the individual pieces of their shell that are called coccoliths. These coccoliths will interact with light in such a way that when they reflect light, there is a turquoise color. And so you can see that here in this bloom that of coccolithophores off the coast of Newfoundland, Canada. And just this really pretty turquoise color. And I have another example here that was taken from north of the Bering Strait. So water from just north of where Russia and Alaska almost meet. And there seems to be some coccolithophores on this part of the body of water, and maybe a different group of phytoplankton dominate in some of these other areas where it's greener. And so I mentioned how phytoplankton are really important for exporting organic carbon. So that's carbon that makes up the parts of the cell, so that's the body of the cell. But for phytoplankton like coccolithophores, their shells can also sink out of the deep ocean, out of the surface ocean, make it to the deep, and eventually become incorporated into the rock record. And that's has happened here after millions of years. So these are the White Cliffs of Dover in the UK, which were formed millions of years ago by coccolithophores. So all this rock is calcite or calcium carbonate that is from coccolithophores. Now, looking back at coccolithophores under a microscope, this is an image I took, and you can see that there are 3 mostly complete shells of a coccolithophore. So one here, and one here. And then I just have highlighted the different orientations of some of these individual pieces of the shell that are called coccoliths. And so here's one that's upright. So that's in the position that they are when they're attached to the cell. Here's one that looks like it's upside down. And then here's another one that looks like it's on its side. And as you might imagine, since coccolithophores have this organic part and this inorganic part, they have a unique role in the carbon cycle, right? So because of photosynthesis, they transform carbon into cell matter. So that's the organic carbon part. And because of calcification, they transform carbon into calcium carbonate. And so that's the mineral that forms the shell that surrounds the cell. And so both processes use carbon that's present in the seawater. And of course, there's also the sinking of carbon, both the organic carbon of the cell matter and also the inorganic carbon from the shell that can sink out of the surface ocean into the deep ocean and maybe even the sediments. But it gets even more complicated than this with coccolithophores, because calcification also releases carbon dioxide. Now, here I have it shown as the coccolithophores taken up carbon dioxide for photosynthesis and for calcification. It's a little bit more complicated than that. So carbon can exist in the ocean, inorganic carbon, I should say, can exist in the ocean in a few different states. And so what the kind of carbon that this cell is taken up is most likely bicarbonate. And so they take up bicarbonate for both photosynthesis and calcification. However, coccolithophores release carbon dioxide after calcification. And so this is analogous to carbon dioxide that's in the atmosphere, where there's more carbon dioxide now because of human emissions, then enter in the ocean through air-sea gas exchange and could potentially contribute into ocean acidification. So the idea here is that if there's a lot of calcification, you might have a lot more CO2 in the environment due to coccolithophores. Again, this is a different form of carbon that, for simplicity's sake, I did not relabel because I didn't want to get into carbonate chemistry too much, but I can always answer some more questions if that confused anyone. And then in addition to this, there also are these individual parts of the shell, right? So these coccoliths, and these can become detached from the cell. And these individual coccoliths can interact with other particles in the water column, whether it's detritus from other dead organisms, maybe other phytoplankton, or other coccoliths, and they can come together and help to export carbon out of the surface ocean as well. So, in order to understand how coccolithophores impact the carbon cycle, we have to understand the relationship between photosynthesis and calcification. And so I like to think of it almost as like a seesaw. So coccolithophores are doing both. And what we want to know is what's happening more than the other one. So, right, if you're on a seesaw, if a kid's on a seesaw in a playground, if one person is much heavier, they're going to be down close to the ground and the lighter person will be up. And so if we have photosynthesis happening at much higher levels than calcification, there's going to be a net carbon uptake. So they will be taking more carbon out of the ocean, which means that they are a carbon sink. And so this is the same way that plants can be thought of as a carbon sink, and how plants and trees and such are often used as ways for companies to offset carbon emissions because they protected a forest, for instance, and hence that's a carbon sink. But the reverse can also be true with coccolithophores. We could have more calcification than photosynthesis. This would mean that there's net carbon release. And remember, this is carbon dioxide that's being released, meaning that coccolithophores would be a source for carbon. Right? And so one of the goals of my research is to better understand which of these processes is dominating. That way, we can have a better understanding of their role in the carbon cycle. And so now I'll just mention a few ways in which I can measure photosynthesis and calcification in the lab. So for starters, I grow coccolithophores in the lab. So these are two culture flasks that have coccolithophores in them. And there's probably easily tens of millions of cells in both of these flasks. So these are what we would call pretty dense, at least what I would call pretty dense. And the species that I study has the ability to exist without forming a shell. So this means that all the carbon that we are interested in for this species is all the organic carbon. So all the carbon that is made from photosynthesis. And then I also grow the cells that form the shells, like normal coccolithophores. So I can perform the same measurements and the same experiments on both of these cell types, and that in order to better understand how calcification impacts photosynthesis and the differences between photosynthesis and calcification. So one way that I measure photosynthesis first involves understanding what happens to light when it enters the phytoplankton cell. So when the light reaches the part of the cell where photosynthesis happens, a few things might take place. So there are a few different fates for that light energy. First, surprisingly, it can be used for photosynthesis. That light could also be given off as fluorescence, or it could be used to dissipate excess energy in the form of heat. So in other words, if the cells are receiving too much energy and they can't handle it, they will dump some of that light energy as heat. And so in the lab, we can use an instrument that exposes phytoplankton to light and then measure their fluorescence. And using that, we can then calculate how much photosynthesis is happening. And if we're interested, we can also do the same thing with heat using fluorescence. So that's one way to look at photosynthesis. And now one way that I measure photosynthesis and calcification is I start by growing up cells in a culture flask. And then I can collect the cells onto a filter and then expose them to acid. And this will separate out the cell matter, so the organic carbon from photosynthesis, from the inorganic carbon or the mineral, right? So this is what forms the shell from calcification. And so this will tell me the total amount of organic carbon in a cell and the total amount of inorganic carbon that makes up the shell of a cell. But I can also do this, essentially the same process, but add reactive carbon, or C14. And so with C14, we can use radioactivity to actually track where the carbon ends up. So we are given the cell's carbon to take up, and then we track where that carbon goes. And the end result is that we can directly measure photosynthesis and calcification, right? And we do this and we can calculate this because we can calculate how much organic carbon is being produced over time. So it's a rate, so the rate of organic carbon production. And we can also calculate the rate of inorganic carbon production, right? So the rate at which cell matter is being made and the rate at which calcification is being made. And so now I'll present some of the highlights of my research, sort of the take-home messages, if you will. So I found that calcification leads to enhanced photosynthesis. And that is because in cells that are calcified compared to cells that don't have a shell, there was 10 times the organic carbon production, right? So 10 times more organic matter was made in cells that made a shell compared to cells that did not. So now looking at just the cells that have the shell, so just calcified cells, I found that calcification was about 2 to 4 times greater than photosynthesis. So they were making more inorganic carbon than organic carbon. And so if we go back to the seesaw analogy here, I found that calcification was greater than photosynthesis. And so that suggests that they may be a source for carbon, right? So there might be a net release of CO2. But I also did those measurements of just how much organic carbon is in the cell and how much inorganic carbon is in the shell. And those measurements showed that cells have more organic carbon than the mineral. And so this might be a little confusing. So I try to think of a way to make this easier to understand by thinking of it as balancing carbon accounts. So there's more calcification than photosynthesis, but there's more cell matter than mineral. And so if we think of it like balancing a bank account or a bank account balance, we have the mineral and the cell matter that are the final balances that you see when you look at your bank account. So what I found is that the cell matter has a higher balance than the mineral. But I also measured the income. And so calcification, the income for calcification was greater than the income for photosynthesis. But in order for the balance to be like I observed, it suggests that the expenses should be greater for calcification than they are for photosynthesis. And so that leads to the question of, well, what happens to this extra calcium carbonate that disappears, right? So if it's an expense, that means something's happening to it. That way we don't measure it. And so one hypothesis we have is that maybe once the coccoliths become detached, so these are the individual pieces of the shell, once they become detached, maybe they're rapidly dissolving in seawater into carbonate. And so this is an active question that we're hoping to investigate with some collaborators at another institution. So I can't say for sure what's happening here, but that's our best guess as of right now. And so I hope I have convinced you that it's important to understand phytoplankton and particularly coccolithophores in order to understand the carbon cycle, right? So there are so many ways that in which coccolithophores impact the carbon cycle. And so in order to understand their net impact, we have to understand what processes are going on both within the cell and outside of the cell. That way we can predict what their global impact is, right? And so my work is a piece of the puzzle to understanding the global carbon cycle. And another reason why this is important is that, as I mentioned at the beginning, we are changing the carbon cycle by putting more carbon into the atmosphere. And because of that, the Earth is also warming. So ecosystems are going to shift to respond to climate change over the next century and beyond. And so in order to be able to better predict and understand how things might change, we still need to better understand what's going on right now. And yes, it's changing right now, but it'll change more in the future. And so hopefully I didn't lose too many people through that. But in case I did, I thought I would end it with some talk about field work that is often more exciting. And so scientists use a bunch of different tools to study the ocean. And so as you may have guessed, satellites are commonly used. So a lot of the data from the videos I showed earlier, as well as the images of blooms, were collected via satellites. And this is how we observed blooms of phytoplankton and the reflection of light from coccolithophores. So that makes us aware that it's a coccolithophore bloom. There's also different floats and autonomous gliders that can be placed in the water to measure certain attributes, either chemical, physical, or biological of the water column. And then, of course, there's the good old-fashioned fun way of you can go out to sea on a ship and throw a bunch of things over the side. And by things, I mean complex scientific equipment and instruments that measure specific things or that collect seawater that then you can bring up and play around with in the lab. So I recently got to go out to sea earlier this year on a research cruise that was funded by the National Science Foundation, or NSF. And so the ship, the RV Hugh R. Sharp, its home port is in Delaware at their Earth, Ocean, and Environment campus. And the goal of this cruise was to better understand particle disaggregation. So I mentioned particle aggregation, where pieces of dead organic matter can come together and potentially enhance the sinking speed of those particles. But the reverse can also happen because they're always being decomposed and preyed upon by other microbes. And so they can also break apart. So that was the goal of this cruise. And we wanted to go into the Gulf of Maine. That's circled there in red, and get a few spots that were out in towards the open ocean, towards the edge of it, or like right here by the east side, and also some more coastal regions pretty close to coast of Massachusetts and New Hampshire, with the intent of maybe getting some input from rivers. But Mother Nature had some other ideas, so we were only able to get this far north because the weather ended up being too rough for the ship. But we ended up just hiding from the weather close to the coast of Massachusetts, and we were actually in Narragansett Bay in Rhode Island. And then on our way back, we were able to stop in the Hudson Canyon, which is a very deep canyon that is where the Hudson River used to dump out at during the last ice age, before we then returned to port. So now I have a few videos and images to show just to conclude the talk. And so this profiler on the left here is a CTD, and it is placed into the water, and it has instruments, sensors down at the bottom that collects data like salinity, temperature, depth, as well as chlorophyll fluorescence, which is then how we know how much phytoplankton are there. And it will then descend into the water, and it will look like this just before it's about to go beneath the surface. So you can tell it's going down, since all of the tops of these bottles are still open. And so now this video below will show what it looks like, a side view of it from the back of the ship when it comes out of the water, and the boat's rocking and it's going to come up to the crane. So it's just going up. I really like this video because it really showed how massive the crane was, and also just how well the whole system worked. So on other ships, you have to use a line to manually tag the CTD as it comes out. On this ship, we didn't have to, just because of their system. And so it's as it's coming out, the boat's still rocking and everything. So everyone, needs to keep their distance, but then eventually it'll come back in. So this is what it looks like up close. So here's what the top of the bottles look like, as well as the bottoms, along with the other sensors and such. And this is just the inside of a bottle, in case you were curious. And so on the ship, when it's in the water, we can tell the bottles to close at a specific time. So we can tell, say, 3 bottles, you're going to close at 10 meters, and the next 3 bottles, you're going to close at 20 meters, et cetera, et cetera. So we can get water from different depths just by deploying it into the ocean once. And then once it's back up, the job of people like me who are interested in playing around with what's in the seawater is to collect the seawater from the bottles, which is what I was doing here, and then go into the lab and play with it and do what we have to do. So I was mostly filter in the seawater. That way we could then analyze the filters later in the lab. And so this video here is of the water being filtered, and there's little copepods swimming around. So this is so that's zooplankton. And I'll play that again since it's pretty quick. And this is actually the plankton that Plankton from SpongeBob is based off of, which lots of kids typically know who that is. And of course, the entire time that we're on the ship, working in the lab or out on the deck, the ship's rocking. So the top video, you can see, hopefully as long as Zoom isn't buffering, how the water in the filter towers are moving, rocking everything. And then you can just see, based on the angle of everything, how the ship's rocking. And since we were avoiding some bad weather, we did feel some heavier waves. And of course, one of the best parts about being out at sea for someone like me who mostly does lab work is that in addition to doing the lab work in the labs on the ship, you can also get out under the deck and just help put other stuff in the water and such. So these are just some images of me helping out with a bunch of other profilers that other members of the science crew were interested in measuring different characteristics and parameters of the water. And so I'll just end with some acknowledgements. So both of funders of my work, and then of course the Phyto and the Temachichrome labs that I'm a part of. Liti is a part of these labs too, just in case you didn't know. So she gets thanks for helping to with a bunch of technical questions along the way. And then the rest of the students in my program, the department as a whole, and since I talked about the crews at the end, the crew of the ship, as well as the other scientists who were on the cruise. So I will take any questions, whether it's about the science, sort of how to become a scientist, or anything else. I'm happy to take questions.
Hi, thank you so much, Austin. That was really interesting. I seem to have asked the most questions, but I'm going to start with one that I didn't put in the chat, which is, oh, hold on, wrong thing, which is from Yvette Schlessel, who is on our sustainability task force. And she asked if, okay, let me find it.
Is sewage
dumped
okay.
in the ocean responsible for more algae blooms?
Yes. So pollution from human sources can lead to more algal blooms, and especially harmful algal blooms, or HABs, that often make the news because of, I mean, they're harmful, so they typically can lead to things like fish kills and other bad things for the ecosystems. Sometimes they're even toxic species that can be detrimental to human life. And so the important thing to remember is that essentially phytoplankton are plants. And I guess I had this in the talk, then I cut it out, but I'll mention it here now. So algae and phytoplankton are overlap, but they're not identical. So phytoplankton is, right, basically if you're microalgae, so unicellular or a small colony, that you're a free drifter. So you're going to go with the currents of the ocean or of the river if it's in a freshwater ecosystem. Then you're phytoplankton and you're microalgae. I mentioned earlier on when I was at Susquehanna, I worked with algae that grew on surfaces. So even though they were still diatoms, which can be phytoplankton, since they grew on surfaces and not in the currents, at least in that stage of their life, they would be not be considered phytoplankton. So they're algae, but they are more, the terms can more or less be used interchangeably, but there are some differences around the edges. So since they are basically plants, they require the same nutrients that that plants do. So we think of fertilizers that are applied to fields. They typically contain some source of nitrogen and some source of phosphorus. And these are the two big nutrients that phytoplankton need too. And whether it's a freshwater phytoplankton or algae or marine, they will need both. In the ocean, nitrogen tends to be limited, while in freshwater ecosystems, phosphorus tends to be limited. But if you have a huge dump of water with lots of fertilizer or sewage, since that can serve as the same thing, it can potentially lead to more algal blooms. So I think I just saw a headline about the big bloom in one of the lakes in California, and they think one thing that helped cause it was, I think, a failure in a wastewater plant that dumps sewage. So short answer is yes.
Interesting. I asked some of the other questions. I'm curious, when you talked about the balance between carbon release and photosynthesis, what causes that to change? Is it just a natural happening, or are there factors that make the balance go one way or the other? And which balance is better? Is it better to be evenly balanced, or one more prominent than the other?
Yeah. So part of the goal of my research is to try to understand what can affect that balance. So I didn't include it here, but I also study these coccolithophores at different light levels. So at light that's really intense, like at the surface of the ocean, and light that's pretty low. So that's at like the bottom of the zone of the ocean where photosynthesis would take place. And my work interestingly found that calcification was greater than photosynthesis at every light level. So that 2 to 4 range was across the light level. It's highest in the middle, but so that's one thing that can change, because they are both dependent on light, since the energy for calcification will ultimately come from photosynthesis. And there's some hypotheses about maybe it's a place for cells to dump some extra energy, but we haven't really found too much data to support that. But there are other factors that are pretty well known to increase calcification. So I was talking about some nutrients that they require, like nitrogen and like phosphorus. And if you don't give them as much phosphorus, it's known that they will increase how much they calcify. So that would probably, assuming it doesn't impact growth, which it often doesn't impact how fast they grow, that would increase how much calcification happens. And so and there are other things like potentially temperature and other environmental factors that could impact it. Typically, organisms like phytoplankton will divide faster at higher temperatures, as long as it's within the range of temperatures that they like. Eventually, you can get too hot or too cold that they will die, but within their comfortable thermal boundaries, they tend to grow faster on the higher end. And
Go ahead, I'm sorry.
I was just going to say, and so which is better? That's probably applying like a human definition of better onto it. And so, in the sense of sort of, I grounded it in the carbon cycle and in climate change. So in terms of that, we would want them to take up more carbon through photosynthesis and export that carbon to the deep ocean to try to store away some of the carbon that we've released out. Whereas if calcification predominates, they'll be also releasing carbon dioxide. So from that perspective, the more photosynthesis would be better. Also, I guess you could say if the object of a cell, if the goal of the cell, quote unquote, is to try to gather as much energy as possible and divide as much, that way they can just spread their footprint in the ocean, then it would make sense that they would want photosynthesis to outweigh calcification. But there were other benefits to calcification that I didn't have time to go into since it's not a focus of my research. But having a shell can help protect you from being eaten and can help protect you from potentially from viral infection. So a lot of the work in my lab by other people focuses on viral infection. And the examples with the animal plankton, with the zooplankton, show that when they eat cells that form a shell, they aren't able to digest it as well as if they just eat cells that don't have a shell. And so it's a balance for the cells too, because they want to be able to grow and divide as quickly as possible, but they also need to be able to protect themselves. And it definitely seems like calcification is a way that they can do that.
So I'm just going to ask you two more questions. Has global warming, you just sort of touched on it, has global warming of the oceans affected those video maps that you showed us of how during the seasons the different areas of the land and the water change due to vegetation both on land and on water? Has global warming affected that, and how does global warming impact phytoplankton, and what does that mean to the carbon cycle?
Right. So I will say, before I answer that, since it's not my specific area, I'm not up to date on on the latest stuff, but in general, I can say, and I'll say climate change rather than global warming. That way I can talk about changes in pH as well. But in general, yes, global warming is definitely impacting both land plants and phytoplankton. So I'm not sure if in the specific videos that I showed, if you can, if a scientist could go back and analyze specific regions. I suspect they could, because I believe that the biomass of spring blooms in the North Atlantic, so like off the coast of New England and into Canada, that part of the ocean, I believe that there's a spring bloom that always happens, and I believe that the biomass of that bloom has been getting smaller as the environment has warmed, but it's still a very active area of research. But in general, particularly with phytoplankton, but for land plants too, there's going to be a migration of, and this is true for animals as well, a migration of species from the equator to the poles, because right closer to the equator is getting warmer. Everything's getting warmer, but the poles are warming the fastest. And so say it might be easier to imagine with a fish. Say if you're a fish that used to live off the coast of Florida, because that's the ideal habitat for you. That's where your food is. That's where the ideal temperature profile for you is. Well, what temperature that water was in 1990, that same profile of water temperatures is now, say, off the coast of, say, South Carolina now. And odds are some of your prey moved up there too. So if you want to survive, the fish probably moved up with like that. And so phytoplankton, as the base of the food chains, are also migrating. So it seems like the species I study, Emiliania huxleyi, is migrating towards the poles, so closer to the Southern Ocean and closer to the North Pole, as as the ocean warms, the ocean surface warms. And then ocean pH, the decrease in pH due to acidification is, again, it's still an active area of research. There's some studies that suggest that specifically organisms like coccolithophores will be hurt because it can help dissolve their shells. But then there's other work that has shown that they're able to compensate for that. So I'm not quite sold either way, since just keeping it, since if it's important to keep in perspective that coccolithophores evolved millions of years ago, like during the age of the dinosaurs, and they were around then, and carbon dioxide levels were much higher then. So that's important to keep in mind. And also, phytoplankton divide much faster than other animals, so they should be able to adapt faster than other animals. But what will probably happen, in addition to the general trend of migrating towards the poles, is that some groups may be replaced as being the dominant. So right now in the ocean, diatoms tend to be dominant. They tend to be bigger, and they tend to be better at taking up, they tend to be better because they show up first and they're able to suck a bunch of nutrients out of the water and then and store them within themselves. But then as they deplete their nutrients and as the nutrient stock that's available in the water decreases, other organisms typically tend to show up and start to dominate. So coccolithophores tend to replace diatoms, and then other groups come in. So one big hypothesis is that overall diatoms will decrease their biomass, and other groups, like maybe coccolithophores and maybe some of the other groups I showed, might tend to increase their abundance. But since diatoms are bigger overall than the others, there is a hypothesis that the overall biomass of phytoplankton may decrease a bit.
Sorry, very interesting. What I want to say, last question, which Leedy kind of thought was a fun question. And this is for anyone out there who is interested in what you do and maybe going into it, any young person, is what's the difference between like a marine scientist and an oceanographer? And do you have both of them where you work now?
Yeah, so I mentioned at the beginning that I had wanted to be a marine biologist when I was a kid, and now I've mentioned oceanography throughout the talk. So, and specifically, I'm a biological oceanographer. So, I guess let's then start defining oceanography. So oceanography looks at the ocean as a system. So there's the physical aspects of the ocean, so currents, waves, tides, and stuff. There's the chemical aspect of the ocean. So what's in the ocean? What are the salts in the ocean? Carbon that's in the ocean, and different kinds of carbon that's in the ocean, and other elements that are in the ocean, nutrients and stuff. And then biology, which is my area. So things like phytoplankton, zooplankton, and then higher up to bigger animals. And there's also stuff like geological and oceanography, which blends into what we'd call paleoceanography, which is where you can basically dig up sediment cores from beneath the ocean and learn about the past of the ocean using those cores. And so the difference between, like, say, biological oceanography and marine biology would be, say, a marine biology, you would probably be more focused on, say, for instance, studying the organism itself and more focusing on the ecology of the organism and such. So, say people who maybe studied like dolphins or whales, sea turtles, sort of the stuff that gets a lot of people into oceanography, at least biologists, I'd imagine, would tend to more steer on the sides of marine biology because it's more focused on the organism. I, of course, from the talk, I focus on the organisms a lot, but I also think about how they plug into not only the ecosystem, so not only their ecology, but also how they plug into the flow of energy and the flow of nutrients throughout the ocean and how they interact with currents and stuff like that. So I'd say there's overlap, especially between biological oceanography and marine biology. But in general, marine biologists tend to be more focused on the organisms, and oceanographers tend to focus a bit more on the broader picture and how it relates to things like, say, the carbon cycle and the biological pump, whereas a marine biologist probably wouldn't really think in terms of that as much.
Interesting. With a couple of questions, real fast answers because we're running out of time. From Elena Chertaglia, who actually is also a graduate in entomology from the school that you're at, Rutgers School of Environmental and Biological Sciences, and she's on our National Moth Week team. And she asks, she's teaching, she's a professor now at Bergen Community College, and she's asking, what are your post-grad school plans?
So I sort of have two tracks that in mind that I don't see as mutually exclusive. So I'm interested in either going into policy, particularly like at the state level, and of course, mostly focus on environmental stuff, simply because it's a passion of mine. And so either policy or teaching, and I see there's a lot of overlap between the two. Communication skills are really important, whether you're trying to teach students concepts or whether you're trying to speak to other policymakers who haven't taken science since high school. And so the teaching level I would be more interested in would be more like at a small school that I went to, like Susquehanna University, or even like a community college where the focus more is on teaching and is on interaction with students than say at a place for Rutgers, where for the students who are pursuing bachelor's degrees, they might be in classes with, hundreds of students. So there might not be much one-on-one time. And the professors there in sciences are more research focused. So teaching or policy is the short answer.
Ask one more technical question. Elena says, if you want to talk more about a career in community college teaching, let her know. So we can certainly put you in touch with her. She wanted to know if coccolithophores are affected by ocean acidification the way that other marine calcifiers are negatively impacted.
Yes, so I did. So short answer is probably, but they also might be able to adapt better than things like oysters and corals because they are microbes, so they divide much faster. So that's the quick answer.
Okay, she says, so we can only take one more question. So I don't think there's another one that I haven't seen. So I think we can wrap it up now. And I want to say thank you. Also want to say that I'm never going to think of the White Cliffs of Dover the same way again, because now I know how they got formed, which I had no idea. So that was very interesting to me. And I want to say thank you to Austin for coming and sharing your knowledge, your considerable knowledge about phytoplankton and what's going on in our oceans and climate change. It was really interesting and enlightening to me because I've never studied those subjects. So I found it really interesting. And I want to say thank you to everybody who attended tonight. And we hope we'll see you again next month for our program on water quality in East Brunswick. Go to friendsebec.com, sign up as a member, which means we will send you notices about programs like this and all recycling and all the other things that we do. And just want to wish everyone a good night and hope if there are any young people watching that you're having a great first week of school. Good night, everybody. Thank you for coming.